Hello, welcome to this lecture on biomathematics. In this lecture, we will try to do some kind of question answering, like that. What do I mean is some kind of tutorial session. Many of the we learned many things, and this will be like we will be discussing a few questions, and we will try to answer them. And later I will also provide you a set of questions which you can try to solve yourself. So, as you know, mathematics is basically like you will learn it only by doing it. It's like somewhat like swimming, or just like this is true for any other language in some sense. Like as we said in the beginning, mathematics is some kind of a language to write and to express things very quantitatively. Anything that we see in nature could be described using mathematics, and this is some kind of a language. And this is true with any language, that only by using it constantly you will learn it. So, the only way of learning mathematics is basically trying to solve problems, trying to frame problems. So, there are two kinds of things in solving problems. One is you are given some equations and asked to do something like find derivative. Typically, this is the kind of questions that a typical exam will see, like find derivative or find to ask you to do some operation that you have learned. That is the easiest part of it, which is you are taught to do some kind of operation, differentiation, or even in schools you are taught to do multiplication and all that. And again, you might be asked to do some questions like in that way. But if you think of in a more advanced level, what you would need is you will be described the phenomena, some phenomena that we see in nature, and you will have to think of how to write a mathematical expression for that phenomenon, How do you describe, how do you write down that phenomenological information that you got into an equation? That is the biggest challenge. If you can write down that phenomenological information that you got into an equation, then you are successful, then you can solve it. So, here we will do both. We will do some exercises where you will be asked to do some simple operation that you have already learned. You will also be given some description. You will also have some description, and from the description you will have to figure out what is going on and write down some equations or plot that information, present it in a quantitative way. So, that is what we have to do. So, first we will take a question where through this I will describe to you a phenomenon which is a very well known biological phenomenon, but some of you might be hearing this for the first time. Hearing that phenomenon description, first you have to present it in a quantitative way. So, what is the simplest way of presenting something quantitatively? To make a plot. So, you have to make a rough plot of whatever the description you see. So, when I say description, that will be in English, right? It is a language, I describe it. Then when you convert that into a graph, you are doing some kind of transformation from converting the qualitative description to a quantitative information, which we can write down as an equation later. So, and from that we can do various other operations and get many other useful information. So, let us go to the first question. Here the aim is basically, so first there will be a description of a phenomena. You listen the description carefully, understand them, and then you have to plot whatever it is described. So, I will do that, I will do the description and I will do the plotting, and I will take you through how one does this, and later you will have other examples where you have to do it. And then we will also do some very conventional, very typical questions like you have to do some particular plot some function or do some particular operations like find different integral or find derivative or so on and so forth. So, let us first look at this description and let us see how to plot that. So, let us go ahead. So, today's session is tutorial and discussion, where we will be discussing questions as I said. So, the first question is basically is on dynamic instability. So, here is a description. So, we will slowly go through the description. So, I will read it, we will read this carefully and let us see what we understand from this description. So, imagine that somebody is telling you some phenomena that is being seen in biology, or this is that we learn from a lab experiment. Let us say somebody is telling you this description. So, let us go through this carefully. So, microtubules exhibit a phenomenon called dynamic instability. So, microtubules are a particular filaments in biology that you see in cells actually. So, they exhibit a phenomenon called dynamic instability. In this, that is in this phenomenon, first tubulin monomers polymerize and grow with some speed to a microtubule filament of certain length. So, first what happens is tubulin monomers polymerize and grow to a polymer, and the polymer will have a certain length. Then suddenly depolymerize and shrink to a very small filament, then again starts growing, then shrink again. This process of growth followed by sudden shrinkage is repeated. This is called dynamic instability. So, let us understand this once more. So, there is microtubules. So, this is a phenomenon exhibited by a set of filaments in the cell called microtubules, and the name of the phenomenon is dynamic instability. And microtubule is a polymer, it is a long polymer, it is a very stiff polymer, it is like a rigid rod, it is almost like a rod like this pen. Well, imagine that this kind of a pen is made of many subunits, and for example, like I would just draw the picture here. So, you could imagine that many subunits joining together to form a filament, which they have connections like this. So, there are similar, you can imagine microtubule something like this. For simplicity, it is more complex than that. There are 13 protofilaments and so on and so forth, but for the moment you all know that it is a polymer and they polymerize. So, here tubulin monomers polymerize and grow with some speed to a microtubule filament of certain length. So, what happens here is that the filament you start with one monomer, then they polymerize 2, 3, 4. So, in 1 second certain number of monomers are being added and then they polymerize and then grow. So, they just keep growing like this. So, that is the first part. So, microtubules in this first tubulin monomers polymerize and grow with some speed to a microtubule filament of certain length, then suddenly depolymerize and shrink to a very small filament. So, what we saw is basically polymerize and grow. So, basically we saw growing to a small filament like this, then suddenly, so this grew like this, then suddenly at some point it starts to shrink. So, that means it starts to grow this way. So, suddenly it will be, so you have a filament of length this much, suddenly it will become a filament of length this, it will just start shrinking, the length will just decrease. So, that is the second part. Suddenly depolymerize and shrink to a very small filament, then again starts growing, and after some time again starts growing. So, here it might again start growing and then shrink again and grow again and shrink again and grow again. So, if you look at this is a filament, it will just grow, shrink. So, you can think of this way, it can just grow, shrink, grow, shrink. So, the filament will grow, shrink. So, this phenomenon of growth and shrinkage, this is called dynamic instability, and there are a couple of points. One is to note here that it grows with some speed and then suddenly depolymerize and shrink, then again start growing and then shrink again. So, this process of slow growth followed by sudden shrinkage. So, the growth is slow, but the shrinkage is sudden. So, this is another important point here. There is followed by slow growth and sudden shrinkage, and this is repeated, and this is called dynamic instability. Okay, so, we understand the phenomenon now. Now, let us see what are the questions. The first question is basically plot a schematic graph of length versus time of a microtubule filament doing this dynamic instability. So, roughly you can plot a rough graph actually. You plot a rough graph of whatever this description you heard from this description. Can you plot length versus time of microtubule? So, from this what we say here, you know that the length increases then decreases, length increases, decreases. So, it has to. So, what we need is to plot length versus, this is length versus time. So, when we want to plot length versus time, here what we should have is the axis are other way. So, we should plot time in the x axis and length in the y axis, and at t equal to 0 it will have some length, and we know that this is going to be growing and shrinking and growing and shrinking, but there could be one could plot many ways of growing and shrinking, like something is increasing, decreasing, increasing, decreasing. So, one could plot something like this, but is this correct? So, let us look at a question. So, this is here the length is increasing, decreasing, increasing, decreasing, increasing, decreasing. So, that much is correct, but if you look at this question here, it said that there is slow growth and sudden shrinkage. So, the growth is slow and shrinkage is sudden. So, the speed of growth, slow growth means the speed is very small, very low. So, what is speed? Speed is basically how fast it grows. So, if it grows like slowly, the shrinkage has to be faster than that. So, this slope has to be larger than this, it has to be shrink faster. So, taking this into account, one can draw it slightly differently. So, this is time and this is length, and then you start from here and grow slowly and suddenly come down, slow growth, sudden shrinkage, growth, sudden shrinkage, growth, sudden this should be at least perpendicular, sudden shrinkage. So, this is one way of drawing this graph. One could also draw a little more, this drop can be little more faster, slower. So, one can draw also, this is time versus length. So, growth, shrinkage, growth, shrinkage, growth, shrinkage. This particular way there is a growth and shrinkage. So, you can say this shrinkage is faster than this growth. Okay, this is the slope. If you look at the slope here, this will be much larger than this slope. So, that is a typical way. So, typically if you look at various books, for example, one book you should look refer for this is a book by J. Howard. So, this book on motoproteins and cytoskeletal filaments have a very nice description of dynamic instability. There are many books will have it. Of course, the Albert's book of molecular biology of the cell by Bruce Alberts et al. Is a very good book to know the phenomenology, and also the Howard book will give you some kind of a mathematical description also. So, these two books you should look at for this Alberts et al. Molecular biology of the cell. So, this is book name is Alberts et al. Book. This is one book. Second book is Alberts et al. Molecular biology of the cell. I think it's typically it's called cell, right? I think it's called molecular biology of the cell. So, this is Alberts. These two books will have nice description of dynamic instability. So, and you can also see video there in Albert's book, or you could even see it in YouTube, and from those videos it will be clear for you that phenomenon is roughly something like this: very slow growth and nearly sudden shrinkage. Slow growth, sudden shrinkage. Okay, so now this is length and this is time. Now, what do we need here? What is the second question? The second question is also plot the derivative of length versus time graph. You have to plot the derivative of this. So, what do we mean by derivative? So, derivative, let us plot on the same graph here itself. Derivative of this is d L by dt as a function of t. This is what we have to plot, d L by dt by t. Now, if you plot this derivative of this, look at here. This is just a straight line. So, what is the derivative of a straight line? So, typically as you all know, straight line has a equation which is y is equal to m x plus c, we discussed it, and the derivative dy by dx is m, it is a constant. So, the slope of this part of a curve has to be a constant, and if you take this as a constant function, so until this it's a this is so let's draw this points where the slope changes. So, here till this it is one line, then suddenly the slope changes in different line, one line, slope changes, different line, increasing, decreasing. So, this places where the slope changes, let us mark these points. These are the points where the slope changes. So, now if we plot this, up to this it is just one slope. So, let us call this slope m g, this is the growth slope m g. So, this will be the value of m g. So, then it is just a same function till this. Okay, this is m g, and this is now the second part of this is a different slope, and this is actually again another line, but a line like this. So, as you know, if you have m less than 0 for a straight line, if the slope is negative, the line will look like this. So, this is m x plus c with m negative. So, this is y is equal to mx plus c with m less than 0, the slope less than 0. So, this will look like the slope is like this. What does it? What does this mean? If the x increases, y decreases. If y is equal to mx plus c is positive, means as this is m positive and this is m negative. So, here the m is a negative value, but a large negative value. So, we have to also make a negative axis here. So, let me make this as 0. So, let me call this 0 line, right. This is 0 line. So, this is, so now this one has to be less than 0, much below, and this has to be much larger than this. So, somewhere here. So, it will be much better to draw in a different paper. So, it will be some here, somewhere here much below this. So, let me draw this in a different graph. So, let's just quickly draw it in a different graph here. So, what we have here is first length versus time. So, this is length, this is time, and this. Now, what we want is the derivative. The derivative we want to plot. So, we want to plot here the derivative. So, we want to plot dl by dt, and we will mark these points. So, I just draw vertical line so that we can mark this. So, okay. So, now as we said, this part is a straight line, y is equal to mx plus c. So, this is a positive number. So, this is something here, this is one constant, and this value, as we said, can be, let us say, mg, some growth. Now, this is shrinkage, which is negative slope is negative, but much larger than this. So, it is like this should be much larger than here somewhere. This distance and this distance from here to here, this is 0. So, this is 0, and how much negative it goes from 0, this is minus mg. Sorry, let me call this m shrinkage, ms, which is a negative value. So, up to this, between this line and this line, what we have is something with a large negative value. So, here is large negative value, and here it is again positive value, because it is again same slope as this, then again shrinkage, some slope here. So, you could connect these two, and here again. So, what you will have is something like a square wave here, if you wish. This is the growth rate or the speed of growth, and the speed of shrinkage. So, derivative of this will look like this. So, this is length and this is derivative, and you can know there are only two speeds. If there are many speeds, growth velocity, there are many numbers, you will get slightly differently. You will get this here. You could in principle also get, if the growth and shrinkage speeds differ in each attempt, you could also get something like this, something like the following. You could also get, let us say, dl by dt as is t, and this is your 0 line, this is 0, and you could also get something like to go to different values. So, this growth value here is different, here the speed of growth is different, then this is different from this. So, actually I should draw all these vertical lines with dots, because they are not real So, this kind of way you can draw. So, but you should think that the shrinkage time is slow little, because it shrinks very fast, and then this is much larger than this. So, this you could have this kind of dl by dt versus t graph. Basically, this means that the speed of growth in each attempt is different, and so is the shrinking rate. So, essentially this is one way of drawing this graph, and from this you could learn many things about the rate of growth and so on and so forth, and we could get many quantities. But what we learned here is, from a description, how do we make a graph? So, talking about this, drawing a function and its derivative, you should have like one another interesting question which you also should know is basically, for example, let us think of y and x, and let me draw a function which is this particular way. So, the function is, so let me call this function has some particular value a here and some other particular value b here. It's constant values, and between that it's a straight line. And just below that, here we have to draw the derivative of this. So, we have to draw here dy by dx versus x. So, you can see here, so this curve has three parts. So, here in this part, it's like it's a constant line. It's like y is equal to a, it's a constant. Here is also a constant, y is equal to b, and in between is a straight line. So, you have it's three functions. So, the question to you is that by looking at this without doing any calculation, can you draw the derivative? So, again look at the graph here. So, just by looking at it without doing any calculation, can you draw the derivative here? So, you see there are three parts for this. So, immediately you realize that for this part it is a constant, and when it is a constant, so let us do some calculations in the margins here, right. First it is a constant. So, this is y is equal to 2 or y is equal to 3, it is like a constant, and the derivative of that dy by d2 dy by dx is 0 for a constant. So, here the slope has to be 0. Here it is a straight line, the slope was a constant, some number m. Here again it is a constant, so the slope is 0. So, 0 constant 0. So, it has to be, so let us mark this till this the derivative is 0, till this the derivative is, between these two the derivative is a constant m, and here again the derivative is 0 after this. So, let us try to draw this. Here the derivative is 0. So, let us draw 0 here, and here to here the derivative is a constant. So, let me draw a constant line here. So, let me draw a constant line here, and again it is 0. So, this is 0. So, this looks like a box, like a function. So, the answer looks like, if we have something like this, the derivative of this is 0 here, then it is a constant till this, and then again 0 from there. So, the answer will look something like this. So, this is your function and this is derivative. So, just by looking at it, you should get training to plot the derivatives, and this will help you immediately thinking about derivative without really doing the differentiation by hand, or by learning. Just by physically looking at it, you could make a lot of sense out of many of the things that you see. So, this is the aim of this training. We will also see couple of some questions, which is basically some other functions to plot quickly, because if you can think of functions as graphs, that will surely help you. So, let us look here the next question in the tutorial, which is plot a rough sketch of the two following functions in the same graph. So, y is equal to sin x, y is equal to sin square x. Both of them we should plot in the same graph. So, we have two functions, y is equal to sin x and y is equal to sin square x. So, when we say sin x, we immediately know how does this graph look like. So, the sin x typically, so let me draw this a little more carefully here in a different plot. So, sin x typically will look like, let me start from here. Look like starting from 0 is like a oscillating function. So, and this, so when you say plot, you have to know the, so at 0 it's 0, and again sine, the maximum value of sine has to be plus 1 and the minimum value here has to be minus 1. So, let us mark this, and here this becomes, this point is basically pi by 2 and this is pi, this is 3 pi by 2, this is 2 pi. So, 2 pi, one cycle is completed and reached back, and then everything repeats. So, this is you add pi by 2 and just keep going this way. So, this is y is equal to sin x, the blue curve. Now, what I will show here in red is y is equal to sin square x. So, now this is our 0, sin x goes from minus 1 to plus 1. Now, when, so this is y is equal to, in the blue, what is we show here in blue is y is equal to sin x, and this is our x and this is our y. Now, when it is sin square x, so what we want to plot is basically y is equal to sin square x. So, this is square. So, if something is square, it always has to be positive. So, whatever sin x is the square of this. So, whatever I am going to plot is only going to be above this, because it is square of something, sin square of sin square. So, it is only positive. Anything square is only positive, even minus something square is positive. That is first thing we learn, it has to be above this. Second, see here the maximum values are plus 1 and minus 1. So, the maximum values of the square also has to be 1, because 1 square is 1, minus 1 square is also 1. So, the maximum value has to be 1. But all the values here, so we know that we immediately we can say sin square x at this point will be the same, because 1 square is 1, and minus 1 here square is also 1. So, these points, this is going to be the same value here, same values. This is plus 1, value is plus 1. Now, here for any other value, for example, these values, the square of this is basically less than, this is less than 1, this red spot here is below 1. So, square of this, let's, for example, let us take 0.5, and square of this is 0.25, which is less than 0.5. If this was 0.5, square of this will be below this, because less than 0.5. So, anything less than 1 square has to be below this. So, the curve all over here has to go below this and hit here, and come below the 0, it should hit 0 because 0 square is 0. So, it should hit here, come back, hit here, come back, hit here and come back. So, it has to go smoothly below this and hit the curve the same. So, this sin square x roughly will look like this. So, if you ask to plot sin x and sin square x, so sin square x will roughly look like, sorry, this has to be 1 everywhere. So, the height has to be the same, this has to be 1 everywhere nicely smoothly 1, it has to hit 1. So, smoothly between 0 and 1 it will go. So, the curve look like this. So, do plot this yourself. So, this is sin square x, sin square x versus x. So, plot yourself and learn this how to plot it, but this is an interesting exercise that you can do. Okay. So, the next question basically is find the derivative of the following series. So, we learn many series, and for example, we learn e power x and so on and so forth. We even learned x sin x cos x etc. in terms of series. So, this is here is a series log 1 plus x, and you have to find out the derivative of this, and the derivative of this will turn out to be one of the series that you already learned. So, do this yourself. I am just leaving it as a question to you to do this and try this as an exercise, and it will turn out that the answer is something that you already learned, one of the series that we discussed. So, do this yourself, and we will just go to the next question, and if there is time we will come back and do the answer, we will answer this. But now we will go and look at another function, another interesting function to plot, which is very much used in biology. And see, this is again something we learned is to schematically plot functions. So, we will learn this function so that we can plot it. We will learn, we will do an exercise, we will do a tutorial of plotting a function in a very systematic way, in a systematic way, so that this will be an exercise for that. So, let us go to the next question. So, the question is the following: in biology, typically enzyme kinetics is described using the Michaelis-Menten equation. So, this is this kind of an equation. Most of you have seen the Michaelis-Menten equation, which is V of s is Vm_s by Km plus s, and this is basically the enzyme kinetics speed velocity, and s is basically related to the substrate, and Vm is the maximum speed, and so on and so forth. It does not matter whether you know this phenomenon or not at this moment, but most of you know, most of you have learned a bit of biology, would know this relation. This is an equation in enzyme kinetics known as the Michaelis-Menten equation, and we have to schematically, systematically plot this function. Most of you know how to plot, how does it look like, but let us use all this what we learned and then systematically plot this function. So, we discussed a set of rules. So, let us look at the function what we have. We have V of s is Vm_s by Km plus s. So, this is the function that we have, and we want to plot it. So, whenever we want to plot it, so typically s will go from 0 to infinity. So, 0 to very large number. So, s is equal to 0, let us say it goes to s is equal to infinity. So, that is, if you plot here this s from 0 to infinity, we have to plot V of s. So, the first thing we should know is that what is the value of this at s is equal to 0 and at s is equal to infinity. So, let us look at the function at s is equal to 0 and s is equal to infinity. So, let us quickly look at this. So, our function is v, again v of s is v m s divided by k m plus s, and at s is equal to 0, we have v m into 0. So, we have v m into 0 is 0 in the numerator, plus k m plus 0, which is k m, 0 by a constant, which is 0. So, at s equal to 0, the function is 0. So, in this plot here, it is 0. Now, let us look at far away, s is equal to infinity, what happens. So, now at s is equal to infinity, as s goes to infinity. So, let us look at again here, we have v is v m s by k m plus s. There is large, if you just substitute s is equal to infinity, this will be infinity by infinity, because yes, this will be infinity here, this is also infinity, because infinity plus k m is also infinity, and infinity into v m is also infinity. Another important point that you should know is that v m and k m are positive constants for the moment. V m is something 0 greater than 0, and k m is something greater than 0. So, this is something which is given to you, and then we have infinity by infinity. So, now whenever we have such infinity to infinity, the rule is something which we discussed called L'Hospital rule or L'Hopital rule, which is basically to find the derivative numerator and denominator, and then substitute the value of s. So, if you find the derivative in the numerator, which is derivative of v m s with respect to s, what you would end up is just derivative of v m s with respect. So, d by ds of v m s is just v m, this is derivative of numerator, and derivative of denominator, which is derivative of k m plus s, is basically 1, because derivative of k m is 0 and derivative of s is 1. So, derivative of this has to be 1 in this particular way. So, we have if you substitute here, the answer we get is basically v m by 1. So, the answer is going to be a constant, which is v m. So, the answer is basically v m. So, at s is equal to infinity, s going to infinity, v of s will go to v m, the maximum velocity. So, in this graph that we have to plot, here the value is here, and for a large value of s, the function has to have some value v m. Starting from here, it has to end here. So, it has to have some curve between this value and this value, and the only question is there any maxima or minima or not. So, the next rule that we learned is to find out the first derivative and then see if there is any maximum or minima or a maxima. So, what we need to find out is to find out the derivative of this function. So, we have to, as the rules we learned, we have to find out the derivative of this function. So, we have this function v of s is v m s by k m plus s. So, let us find the derivative, which is dv by ds, which is basically d by ds of the numerator, which is v m s, plus, so minus, and you have v m s into derivative of, so actually you have plus v m s into derivative of, let us apply this u by, so let us apply u by v rule. This is like u and v, this is like u by v rule. So, you have to find the derivative of the numerator, multiply this with y 1 by k m plus s, then 1 by Km plus S, then we have to multiply Vms into derivative of 1 by Km plus S. This is what you have to do. So, let me write this a little more carefully. So, what we have, this is the u by v rule. So, you can write our function which is v of s as Vms by Km plus S, dv by ds is, you can write as d by ds of Vms into Km plus S whole power minus 1, which is basically Vm. So, now we have to find, let us find the derivative of this whole thing, which is basically derivative of this into Km plus S whole power minus 1 plus Vms into derivative of Km plus S whole power minus 1. So, now what is the derivative of this, which is a constant, which is basically Vm, and you have 1 Km plus S inverse, which is Km plus S here, and then you have there is a derivative of something power minus 1 will give a minus, and there you have Vms, and this will have 1 by Km plus S whole square, because minus 1 will have a derivative which is minus 2, which is 1 over 2 into derivative of this, which is 1. So, this is what you will get, and we want this, we want to find out for any value of s, will this be 0. If the derivative is 0, that means there is a maxima or a minima. There is a maximum or a minimum, at least one. So, let us see if the derivative is 0 at all. So, let us equate this to 0. So, what we have here is basically, if you go from here to here, Vm by Km plus S has to be equal to Vms by Km plus S whole square. So, here there is Km plus S whole square. So, this 1 Km plus S here and 1 Km plus S cancels, and 1 Vm cancels. So, you have 1 Km plus S cancelling here with 1 Km plus S, and Vm cancelling. So, what we have here in the left hand side, this is 1. So, basically you have 1 is equal to S by Km plus S. In other words, if you rewrite this, what does this mean? This means that Km plus S equal to S. For any value, if Km is greater than 0, this will not be true at all. If Km is 0, which is given, this can never be true. So, therefore, there is no such minima exist. So, this dv by ds cannot be 0, because this cannot be true. We know that Km is greater than 0, and if dv by ds is 0, this has to be true, and we know that this cannot be true. This implies that no minimum or maximum. The derivative cannot be 0. So, we had this function which we saw had value here at 0, and for large value of this is s here and here v here, and large for small value of x we got 0, and for large value of x we got Vm, and the curve is a smooth curve between these two things. So, just going and then saturating like this. So, for as s tends to infinity, this has to saturate to this value Vm, which we have here, and there is no minima or maxima, is just curve like this. So, you know that enzyme kinetics curve look roughly like this. So, this is slowly increasing and saturating to a constant value. So, this is what we saw. So, we had a few questions and of different flavors, and we had discussion of how to answer them. Now, as a separate file, I will provide some other questions which you can answer, but in this tutorial we discussed a set of questions, and we will have other tutorials for other topics which we will discuss. Okay. So, with this we will stop today's lecture, and we will continue with a discussion of another topic tomorrow. Bye.